Hello everyone, my name is Bibek Samanta, and today I'm going to present to you some of our work looking into the photochemistry of pyruvic acid. This work was a collaboration between the Reisler Research Group and David Osborne, and I would also like to thank the Advanced Light Source Synchrotron for the resources and DOE for the funding. In this talk, I have highlighted why pyruvic acid is a good prototype molecule to extend our understanding of keto acids in the atmosphere, and I've divided our results into two parts. The first part focuses on the S1 state where hydrogen tunneling plays an important role, and the second part deals with the higher excited states where the molecule fragments and explodes. Now, pyruvic acid originates mainly from plant sources and is present in the lower atmosphere, but what makes it unique is that it's lost mainly through photodissociation and not chemical reactions, unlike some other VOCs. This is because the first excited state absorption spectrum overlaps with the solar flux. This band is an n-pi star-type transition, and traditionally in ketones, this excitation leads to a C-C bond fission, also known as Norrish type 1 reaction. However, in pyruvic acid, in the most stable conformer, which is stabilized through hydrogen bonding, this hydrogen can now hop over to the electron deficient oxygen, also lead to OH bond weakening. Now, this entire process leads to an extremely reactive intermediate known as methylhydroxycarbene. Until date, only its most stable conformer, acetaldehyde, has been observed. Now, this is where our experiments in the molecular beam would offer a fresh perspective looking to the primary products to fill some of our fundamental gaps in the whole understanding of the process. So one of the techniques that we used is the multiplex photoionization mass spectrometer, where we initiate the photochemistry in a flow reactor, and in the end we obtain a 3D representation of the whole process, from which we can extract not just mass spectrum and look at chemical species, how they evolve as a function of time, but we can also characterize them using their photoionization spectrum. The expected dissociation in the S1 state, which we access using a 351 nanometer excimer laser, unfortunately gives both mass 44 products with empirical formula C2H4O and CO2. So we have also synthesized a monodeuteurated product to confirm our results. As expected, the mass spectrum shows only master charge 44 as the major product peak, and we confirm that using the photoionization spectrum like you see for carbon dioxide in the bottom panel. For C2H4O, however, we see not just acetaldehyde but also vinyl alcohol in a 2:1 ratio. Now, this is already a big deviation from previous results because this is the first time the vinyl alcohol tautomer has been observed in this process. While this serves as an indirect evidence for the formation of methylhydroxycarbene, which once formed can isomerize to both the thermodynamically favored acetaldehyde and the kinetically favored vinyl alcohol, we could also directly observe a transient species with the correct mass for both D0 and D1 methylhydroxycarbene, whose photoionization spectrum happened to line up pretty well with our calculated ionization energy. You would also notice that the lifetime of the D1 methylhydroxycarbene is slightly longer than that for D0 MHC, and that's because deuterium tunneling takes slightly longer than that for hydrogen. And now I would emphasize here that this is the very first time all three tautomers, including methylhydroxycarbene, has been observed in a photolysis reaction. And not just that, we also observed its gas phase chemical reaction, which we describe in more detail in our very recent publication here, which I urge you to check out. Now, I would point out here that recent calculations attest to the existence of a small barrier to hydrogen transfer before decarboxylation can take place in the S1 state. And because our excitation energy is just slightly below this barrier, we are now in the tunneling regime. So another interesting observation resulting from a slower deuterium tunneling is the fact that we also observe now this close-lying barrierless Norrish type 1 C-C bond fission reaction, but only for the monodeuteurated pyruvic acid, resulting in CS3CO and DOCO. So now you can already start to think that the next logical step would be to evaluate these competing pathways as a function of the excitation energy. Which brings us to part 2, where the excitation energy is now almost double, and now we're exciting the pi star band. At this energy, we see several products which we confirm using their photoionization spectrum, and I show you a few examples. For example, the OH radical, CH3, ketene, and of course we also see acetaldehyde and vinyl alcohol. Now, using precisely calibrated gas mixture and the known ionization cross sections, we can convert the ion counts for both stable and transient species into absolute product yields. Now, you would notice that at 193 nanometer now, acetaldehyde and vinyl alcohol, which were the major products at 351 nanometer, are only minor here. Now, I think I've already established that the MPIMS technique is an excellent resource as a universal product detector, but what it lacks is the energetics and dynamics, for which we now turn to velocity map imaging, or VMI. I show one example of the kinetic energy distribution of the hydrogen fragments obtained from this dissociation process. We could model the dissociation using a 3-body fragmentation model, which includes the energetics, the dissociation geometry, and the timescales. And doing so, we could infer if, say, a particular process was a 3-body synchronous dissociation, or if it was a 2-step sequential process. And if we now add our findings of these fragmentation processes, we can start to finally fit the product yields with the participating pathways with a certain degree of approximation. And so what we can then conclude is that the Norris type 1 pathway can now compete with the decarboxylation pathway almost in equal amounts, and that both pathways do exist even at higher excitation energies. But now with all the excess energy, there's further fragmentation in both pathways, leading to these many body fragmentation behavior. And so to summarize it all, we see pyruvic acid exhibits both decarboxylation and keto-like fragmentation under different excitation energies, and in fact gives rise to several stable and unstable products, just like the methylhydroxycarbene, which under atmospheric conditions should be further stabilized. And therefore it should be considered in atmospheric modeling along with its tautomers. We have the full version in our most recent publications, which I urge you to check out. And with that, thank you and stay safe.